Hello students, let's focus on urea cycle and its regulation. First, introduction. If not reused for the synthesis of new amino acids or other nitrogenous products, amino groups are channeled into a single excretory end product. Most aquatic species, such as the bony fishes, are ammonotelic, excreting amino nitrogen as ammonia. The toxic ammonia is simply diluted in the surrounding water. Terrestrial animals require pathways for nitrogen excretion that minimize toxicity and water loss. Most terrestrial animals are ureotelic, excreting amino nitrogen in the form of urea. Birds and reptiles are uricotelic, excreting amino nitrogen as uric acid. Plants recycle virtually all amino groups, and nitrogen excretion occurs only under very unusual circumstances. In ureotelic organisms, the ammonia deposited in the mitochondria of hepatocytes is converted to urea in the urea cycle. This urea production occurs almost exclusively in the liver and is the fate of most of the ammonia channeled there. The urea passes into the bloodstream and thus to the kidneys and is excreted into the urine. The production of urea now becomes the focus of our discussion. The urea cycle begins inside liver mitochondria, but 3 of the subsequent steps takes place in the cytosol. The cycle thus spans 2 cellular compartments. First, synthesis of carbamyl phosphate and transformation of ornithine into citrulline. The first amino group to enter the urea cycle is derived from ammonia in the mitochondrial matrix ammonium, arising from the deamination of glutamate by glutamate dehydrogenase. The liver also receives some ammonia via the portal vein from the intestine from the bacterial oxidation of amino acids. The ammonium generated in liver mitochondria is immediately used together with carbon dioxide as bicarbonate produced by mitochondrial respiration to form carbamyl phosphate in the matrix. This ATP-dependent reaction is catalyzed by carbamyl phosphate synthetase 1, which is a regulatory enzyme and a derivative of glutamate. Acetylglutamate is the allosteric activator of this carbamyl phosphate synthetase. The mitochondrial form of the enzyme is distinct from the cytosolic 2 form, which has a separate function in pyrimidine biosynthesis. The carbamyl phosphate, which functions as an activated carbamyl group donor, now enters the urea cycle. The cycle has 4 enzymatic steps. First, carbamyl phosphate reacts with the delta amino group of ornithine, donates its carbamyl group to ornithine to form citrulline with the release of Pi. Ornithine plays a role resembling that of oxaloacetate in the citric acid cycle, accepting material at its turn of the cycle. The reaction is catalyzed by ornithine transcarbamylase and the citrulline passes from the mitochondria to cytosol. Next, we have transformation of citrulline into arginine. This transformation requires 2 steps. In the first step, the second amino group enters from aspartate generated in mitochondria by transamination and transported into the cytosol. By condensation reaction between the amino group of aspartate and the urido group of citrulline, forming argininosuccinate. This cytosolic reaction, catalyzed by argininosuccinate synthetase, requires ATP and proceeds through a citrulyl AMP intermediate. In the second reaction, the argininosuccinate is then cleaved by argininosuccinase to form free arginine and fumarate, the latter entering mitochondria to join the pool of citric acid cycle intermediates. This is the only reversible step in the urea cycle. Next, we have transformation of arginine into ornithine and urea. In the last reaction of the urea cycle, the cytosolic enzyme arginase cleaves arginine to yield urea and ornithine. Ornithine is transported into the mitochondrion to initiate another round of urea cycle. The enzymes of many metabolic pathways are clustered, with the product of one enzyme reaction being channeled directly to the next enzyme in the pathway. In the urea cycle also, the mitochondrial and cytosolic enzymes seem to be clustered in this way. The citrulline transported out of the mitochondrion is not diluted into the general pool of metabolites in the cytosol, but is passed directly to the active site of argininosuccinate synthetase. This channeling between enzymes continues for argininosuccinate, arginine and ornithine. Only urea is released into the general cytosolic pool of metabolites. The citric acid and urea cycle can be linked, as the fumarate produced in the argininosuccinase reaction is also an intermediate of the citric acid cycle. The cycles are in principle interconnected. However, each cycle can operate independently, and communication between them depends on the transport of key intermediates between the mitochondrion and cytosol. Several enzymes of the citric acid cycle, including fumarase, that is fumarate hydratase, and malate dehydrogenase, are also present as isozymes in the cytosol. The fumarate generated in cytosolic arginine synthesis can therefore be converted to malate in the cytosol, and these intermediates can be further metabolized in the cytosol or transported into mitochondria for use in the citric acid cycle. Aspartate formed in mitochondria by transamination between oxaloacetate and glutamate can be transported to the cytosol, where it serves as nitrogen donor in the urea cycle reaction catalyzed by argininosuccinate synthetase. These reactions, making up the aspartate argininosuccinate shunt, provide metabolic links between the separate pathways by which the amino groups and carbon skeletons of amino acids are processed. If we consider the urea cycle in isolation, we see that the synthesis of one molecule of urea requires 4 high energy phosphate groups. 2 ATP molecules are required to make carbamyl phosphate and 1 ATP to make argininosuccinate. The latter ATP undergoes a pyrophosphate cleavage to AMP and PPI, which is hydrolyzed to 2-PI. The overall equation of the urea cycle is 2 molecules of ammonium ion, bicarbonate ion, and 3 molecules of ATP and 1 molecule of water reacts to form urea, 2 ADP, 4 PI, 1 AMP and 2 hydrogen ions. However, the urea cycle also causes a net conversion of oxaloacetate to fumarate via aspartate, and the regeneration of oxaloacetate produces NADH in the malate dehydrogenase reaction. Each NADH molecule can generate up to 2.5 ATP during mitochondrial respiration, greatly reducing the overall energetic course of urea synthesis. The activity of the urea cycle is regulated at 2 levels. The flux of nitrogen through the urea cycle in an individual animal varies with diet. When the dietary intake is primarily protein, the carbon skeletons of amino acids are used for fuel, producing much urea from the excess amino groups. During prolonged starvation, when breakdown of muscle protein begins to supply much of the organism's metabolic energy, urea production also increases substantially. These changes in demand for urea cycle activity are met over the long term by regulation of the rates of synthesis of the 4 urea cycle enzymes and carbamyl phosphate synthetase 1 in the liver. All 5 enzymes are synthesized at higher rates in starving animals and in animals on very high protein diets than in well-fed animals eating primarily carbohydrates and fats. Animals on protein-free diets produce lower levels of urea cycle enzymes. On a shorter time scale, allosteric regulation of at least one key enzyme adjusts the flux through the urea cycle. The first enzyme in the pathway, carbamyl phosphate synthetase 1, is allosterically activated by N-acetyl glutamate, which is synthesized from acetyl-CoA and glutamate by N-acetyl glutamate synthase. In plants and microorganisms, this enzyme catalyzes the first step in the de novo synthesis of arginine from glutamate, but in mammals, N-acetyl glutamate synthase activity in the liver has a purely regulatory function. Mammals lack the other enzymes needed to convert glutamate to arginine. The steady state levels of N-acetyl glutamate are determined by the concentrations of glutamate and acetyl-CoA, which are the substrates for N-acetyl glutamate synthase, and arginine, an activator of N-acetyl glutamate synthase and thus an activator of the urea cycle. Conclusion: the amino group of amino acids can be eliminated in the form of urea through 2 pathways which join in the urea cycle, either by deamination and then incorporation in carbamyl phosphate, or by transamination to aspartate acid. Aspartate acid and fumaric acid establish the link between the urea cycle and the TCA cycle, and the oxaloacetic acid of the TCA cycle can be transaminated and the aspartate acid thus formed enters the urea cycle. The fumaric acid thus formed returns to the TCA cycle. People with genetic defects in urea formation cannot tolerate protein-rich diets. The absence of a urea cycle enzyme can result in hyperammonemia or in the buildup of one or more urea cycle intermediates, depending on the enzyme that is missing. Although the breakdown of amino acids can have serious health consequences in individuals with urea cycle deficiencies, a protein-free diet is not a treatment option. Humans are incapable of synthesizing half of the 20 common amino acids, and these essential amino acids must be provided in the diet.